For thousands of years, the people living across what is now India, Pakistan, and Bangladesh moved, mixed, and settled across this land long before any modern government ever drew a border through it. Scientists compared Indian DNA across Pakistan and Bangladesh, and the results were not what anyone expected. On paper, this should have been one of the more straightforward genetic comparisons in the field. India, Pakistan, and Bangladesh all sit on the same subcontinent, share thousands of years of intertwined history, and were, until relatively recently, part of the same political entity before a series of partitions in the 20th century drew the modern borders separating them. If genetics simply followed national identity, researchers might have expected three fairly distinct genetic clusters, one for each country, each cleanly separated from the others along the same lines drawn on a modern political map. Instead, when scientists actually mapped the DNA of populations across all three nations in detail, using large-scale genome-wide sequencing rather than earlier, more limited marker-based methods, the genetic picture that emerged completely ignored the modern borders separating them, revealing something far older, far more layered, and in several places, genuinely surprising. To understand why this comparison turned out the way it did, you first have to understand something important about how South Asia's population history actually unfolded. Long before any of these modern national borders existed, the Indian subcontinent, encompassing what is now India, Pakistan, Bangladesh, and several neighboring countries, was never populated by a single wave of migration or a single ancestral group. Instead, genetic research has revealed that nearly every population living across this enormous region today descends from a mixture of a small number of deep ancestral source populations, whose proportions vary dramatically from one community to the next, producing a level of internal genetic diversity that few other regions on Earth can match. The most significant of these ancestral components was identified through a landmark study published in the late 2000s, which analyzed genetic data from dozens of populations spread across the Indian subcontinent. Researchers found that virtually every mainland South Asian population, regardless of religion, region, or modern national identity, could be modeled as a mixture of two deeply distinct ancestral populations, which researchers named Ancestral North Indian, often shortened to ANI, and Ancestral South Indian, shortened to ASI. Every population studied carried some combination of both ancestries, but the specific proportion varied enormously depending on region, caste, and community, and this variation turned out to be the single most important genetic pattern shaping the entire subcontinent. The Ancestral North Indian component shows a genetic relationship connecting back toward populations of West Asia, Central Asia, and more distantly, Europe, reflecting a history of migration into the subcontinent from the northwest across many centuries, including later migrations linked to Bronze Age pastoralist populations moving south from the Central Asian steppe. The ancestral South Indian component, by contrast, represents a much older, deeply rooted South Asian population with far less connection to the outside world, and researchers found that its closest surviving genetic relatives today are not found on the mainland subcontinent at all, but among the indigenous population of the Andaman Islands, a small island chain in the Bay of Bengal that remained remarkably isolated from mainland population movements for tens of thousands of years. This gave researchers an extraordinary reference point, an island population that had remained relatively unmixed for an enormous span of time, allowing scientists to model what the deep ancestral South Asian population likely looked like genetically before large-scale mixing with northern populations began. Understanding how researchers actually arrived at this two-population model is itself worth explaining, because the method involved is a good example of how modern population genetics works in practice. Rather than simply looking at broad similarities and differences between populations, researchers used statistical techniques capable of modeling any given individual's genome as a mixture of proportions drawn from a small number of underlying ancestral sources, then tested different possible numbers of ancestral sources against the actual genetic data to see which model best explained the patterns observed across the many populations sampled. When researchers tested this approach across dozens of South Asian populations, a two-source model corresponding to what became known as Ancestral North Indian and Ancestral South Indian consistently provided the best fit to the data, meaning nearly every population's genome could be explained as some specific mixture of these two deep ancestral sources, with the exact proportion varying from population to population in a way that closely tracked known historical and geographic patterns. This gave researchers strong statistical confidence that they had identified something genuinely meaningful about the deep ancestral structure of the subcontinent, rather than simply imposing an arbitrary framework onto the data. Later research, using ancient DNA extracted directly from skeletal remains found across the region, added an additional layer of confidence and nuance to this picture. Ancient genomes recovered from sites across the northern subcontinent and neighboring Central Asia allowed researchers to more precisely date when the mixing between these ancestral populations likely occurred, with much of the evidence pointing towards significant population mixing occurring within roughly the last few thousand years, considerably more recently than some earlier theories about South Asian population history had assumed. This ancient DNA evidence also helped clarify that the ancestral North Indian component itself was not a single simple ancestry, but was already a mixture of earlier ancestral populations, including groups connected to ancient farming populations of the Iranian plateau and groups connected to pastoralist populations that had spread across the Central Asian steppe during the Bronze Age, meaning the full ancestral picture underlying modern South Asian populations actually involves several distinct deep ancestral layers stacked on top of each other rather than simply two. What makes this two-way ancestral model so significant for understanding modern India, Pakistan, and Bangladesh is that essentially no population studied anywhere across the subcontinent turned out to be purely one ancestry or the other. Every community represents its own specific blend, and that blend follows patterns that map far more closely onto ancient migration routes, caste history, and regional geography than onto the modern political borders that now separate these three nations. This question matters for reasons that extend well beyond academic curiosity about ancient migration. South Asia is home to more than 1.5 billion people spread across dozens of distinct linguistic groups, hundreds of endogamous communities, and 3 separate modern nations. And understanding the genetic relationships underlying this staggering diversity has real consequences for medical research, since specific populations across the region carry elevated rates of certain inherited conditions tied directly to their specific ancestral and marital history. It also matters for understanding just how recent and, in genetic terms, how artificial the modern national borders separating these populations really are, a finding that becomes strikingly clear once you look closely at populations living directly along those borders. It also matters for a subtler reason worth mentioning directly, because genetic research of this kind can sometimes be misused or misinterpreted to support nationalist or exclusionary political narratives. It is worth being precise about what this research actually shows and does not show. This body of research does not identify any single population across India, Pakistan, or Bangladesh as more genetically pure, more ancient, or more legitimate than any other. Every population studied across the subcontinent, without exception, turned out to be a mixture of multiple ancestral sources, and the specific proportions of that mixture simply reflect different historical migration and marriage patterns, not any meaningful hierarchy of ancestry. If anything, the clearest lesson from this research is the opposite of a purity narrative, since it demonstrates just how thoroughly mixed the ancestry of essentially every community across the region actually is, once you look closely enough at the underlying genetic data. The first major pattern researchers identified when mapping ANI and ASI ancestry across the region was a broad geographic gradient running roughly from northwest to southeast. Populations in the northwestern parts of the subcontinent, including much of modern Pakistan and northern India, tend to carry a higher proportion of ancestral North Indian ancestry, while populations further south and east, including much of southern India, carry a higher proportion of ancestral South Indian ancestry. This gradient reflects the historical reality that migrations bringing ANI-related ancestry into the subcontinent generally arrived from the northwest, and their genetic influence gradually diminished the further those ancestral populations mixed and spread toward the south and east over the following millennia. But this broad geographic gradient, while real and well documented, turned out to be far less important to the overall genetic structure of the subcontinent than a second factor entirely, one that cuts directly across geography rather than following it. And this is where the research findings became genuinely surprising even to the scientists conducting it. That second factor is the region's long history of endogamy, meaning the practice of marrying only within a specific community, a pattern maintained with extraordinary consistency across many caste and community groups throughout the subcontinent for well over 1,000 years in many documented cases, and likely considerably longer in others. When researchers compared genetic diversity within specific endogamous communities against genetic diversity between different communities, they found a level of internal genetic structure that surprised even specialists in the field. Many individual caste and community groups across the subcontinent showed genetic signatures consistent with a strong founder effect, meaning the modern population descends from a relatively small number of ancestors who founded that specific community many centuries ago, with very little genetic mixing occurring with outside communities in the generations since. Because of the sheer number of distinct endogamous communities across the subcontinent, this research effectively revealed that India alone, before even counting Pakistan and Bangladesh separately, contains more distinct, genetically identifiable founder populations than exist across the entirety of Europe, a finding that reshaped how population geneticists think about genetic diversity across South Asia as a whole. This endogamy-driven structure has real, measurable medical consequences. Specific communities across the subcontinent carry meaningfully elevated rates of particular inherited genetic conditions, a pattern that traces directly back to the founder effect created by centuries of marriage exclusively within a relatively small, defined community. This is the same basic genetic mechanism responsible for elevated disease rates found in other historically endogamous populations studied elsewhere in the world, but the sheer number of distinct endogamous groups across South Asia means this pattern repeats itself many hundreds of times over, each community carrying its own specific genetic signature and, in some cases, its own specific elevated disease risks, shaped by its own particular marital history rather than by any single unified South Asian genetic pattern. This same endogamy-driven structure has produced one of the most well-known and extensively studied examples of founder effect disease anywhere in the world, though not always well understood by the general public, even within the region itself. Certain communities across the subcontinent, shaped by many generations of marriage within a defined social or religious group, show significantly elevated frequencies of specific inherited metabolic and neurological conditions, compared to the general population, conditions that trace back to a small number of founding ancestors who happened to carry a particular genetic variant many centuries ago, long before that variant became concentrated within the modern community through repeated marriage within the same defined group. Public health researchers working across India, Pakistan, and Bangladesh have increasingly used this understanding to develop targeted genetic screening programs designed around a specific community's documented marital history, rather than applying broad, general population screening guidelines that would miss these community-specific risk patterns entirely, since a condition that appears rare across the general population of a country can appear at a dramatically higher, medically significant frequency within one specific endogamous community living inside that same country. Now here is where the comparison across India, Pakistan, and Bangladesh specifically produced results that genuinely surprised researchers. Because when you actually plot genetic ancestry data for populations living directly along the modern borders separating these three countries, the borders themselves become almost invisible in the genetic data. Populations living in Punjab, a historical region split between India and Pakistan during the 1947 partition, show remarkably similar genetic ancestry profiles on both sides of that border, regardless of which country a given community happens to live in today. A Punjabi community on the Pakistani side of the border and a Punjabi community on the Indian side of the border are, in genetic terms, considerably more similar to each other than either group is to more genetically distant populations living within their own respective countries, in regions such as southern India or eastern Bangladesh. Researchers found this same pattern held up when they examined the Punjab data using several completely different analytical approaches, including methods that compare shared segments of DNA inherited from recent common ancestors, a technique that can reveal genetic relatedness occurring within a much more recent historical time frame than the older ANI and ASI mixing signal alone. These recent shared segments showed that Punjabi communities on both sides of the modern border continued intermarrying extensively with each other for centuries, right up until the 1947 partition itself, at which point mass displacement and resettlement abruptly reorganized which side of the new border specific families and communities ended up living on, without meaningfully altering the deep genetic relationships those communities had already built with each other over the preceding centuries. In genetic terms, the partition functioned almost like a sudden administrative reshuffling layered on top of a population structure that had already been fully established long beforehand, rather than an event that created any new, lasting genetic divide between the communities it physically separated. The same pattern repeats itself along the border separating India and Bangladesh, particularly across the Bengal region, which was itself split by the same 1947 partition, before Bangladesh later separated from Pakistan entirely in 1971 following its own war of independence. Bengali populations living in the Indian state of West Bengal show genetic ancestry profiles that closely match Bengali populations living across the border in Bangladesh, sharing not only language and cultural history, but a genetic ancestry signature shaped by many centuries of shared regional history, long before the modern political border between India and Bangladesh existed at all. This cross-border genetic continuity has also been documented in other divided regions across the subcontinent, beyond Punjab and Bengal specifically. Communities living along the border separating Pakistan's Sindh province from India's Rajasthan and Gujarat regions show similarly close genetic relationships across that border, reflecting centuries of shared regional history within a broader cultural and trading zone that existed long before the modern boundary was drawn through it. This pattern holds up consistently enough across multiple different border regions that researchers now generally treat it as one of the clearest, most reliable findings to come out of comparative genetic research across South Asia, namely that genetic ancestry tracks regional and historical settlement patterns far more closely than it tracks any modern national identity, regardless of which specific stretch of border a researcher happens to be studying. What this reveals, and what genuinely surprised many researchers conducting this comparison, is that the genetic structure of South Asia was essentially fully formed by ancient migration, regional settlement patterns, and centuries of endogamous marriage practice long before any of the modern national borders were drawn. The partitions of 1947 and 1971 were significant political and historical events, reshaping governments, populations through mass displacement, and national identities in enormously consequential ways. But they occurred far too recently, within the span of a few generations, to meaningfully reshape the deep genetic structure of the region, a structure that had already been building for thousands of years by the time these modern borders were drawn on a map. Bangladesh specifically adds one further layer of genetic complexity to this picture, one not shared to the same degree by India or Pakistan overall. Because of its geographic position at the eastern edge of the subcontinent, bordering Myanmar and sitting close to Southeast Asia, certain populations within Bangladesh, particularly in regions such as the Chittagong Hill Tracts near the country's southeastern border, show meaningful genetic contributions from East Asian and Southeast Asian ancestral populations, in addition to the broader ANI and ASI ancestry found across the rest of the subcontinent. This reflects a separate and distinct history of migration into the eastern edge of South Asia from Southeast Asia, layered on top of the deeper ANI and ASI ancestral structure shared with the rest of the region, adding yet another ancestral thread into Bangladesh's overall genetic picture that is considerably less pronounced in most of India or Pakistan. This eastern ancestral influence in Bangladesh connects to a much longer regional history of migration and trade, linking the Bengal Delta to Southeast Asia, a relationship shaped by geography, river systems, and maritime trade routes that predate the modern nation of Bangladesh by many centuries. Communities in Bangladesh's southeastern hill regions, in particular, share linguistic and cultural connections with populations across the border in Myanmar and further into Southeast Asia, and the genetic data documenting shared ancestry between these groups closely mirrors that broader cultural and linguistic relationship. This regional pattern illustrates a broader point that applies across the entire comparison between India, Pakistan, and Bangladesh, namely that genetic ancestry tends to follow historical trade routes, migration corridors, and regional cultural spheres far more closely than it follows the boundaries of any single modern nation, since those trade routes and cultural spheres were shaped by geography and gradual historical movement over thousands of years, while the national boundaries themselves were drawn comparatively recently, often along lines determined by political and administrative considerations, rather than by any underlying demographic or genetic reality. Researchers studying Pakistan specifically also identified additional genetic layers not shared uniformly across the rest of the subcontinent, reflecting the country's own distinct position along historical trade and migration routes connecting South Asia to Central Asia, the Iranian Plateau, and the wider Middle East. Certain populations within Pakistan, particularly in the country's northern and western regions, show meaningfully higher proportions of ancestry connecting toward Iranian Plateau and Central Asian populations, compared to most communities further east and south across the broader subcontinent. This reflects Pakistan's long history as a corridor for successive waves of migration and trade, moving between Central Asia, Iran, and the Indian subcontinent, a role shaped by its geography for thousands of years, long before it existed as an independent modern nation. The same corridor role also left a distinctive genetic mark on parts of Pakistan tied specifically to well-documented historical migrations, rather than only ancient prehistoric movements. Communities in parts of northern Pakistan, for example, have long maintained oral traditions connecting their ancestry to soldiers and settlers arriving with historical armies and empires that passed through the region over the past 2,000 years, including forces associated with Alexander the Great's campaigns in the 4th century before the Common Era, as well as later Persian, Central Asian, and Mongol era movements through the same mountainous corridors. All genetic researchers have approached these specific oral traditions with appropriate scientific caution, since many such traditions are difficult to verify precisely using DNA evidence alone, and some claimed connections have not held up well under closer genetic scrutiny. But the broader pattern these traditions point toward, a region that experienced repeated waves of migration and military movement passing through it for thousands of years, each leaving some genetic trace behind, is consistent with what the wider genetic data across Pakistan's northern and western regions actually shows, even when individual community-specific historical claims cannot be confirmed with full certainty. Taken together, this is the central discovery running through the entire comparison. and it is considerably more nuanced than a simple story of three separate national populations. The deep genetic ancestry shaping India, Pakistan, and Bangladesh today was built almost entirely through processes that unfolded over thousands of years, ancient migration bringing ancestral North Indian ancestry into the subcontinent from the northwest, an even older ancestral South Indian population already present before that migration began, additional Southeast Asian ancestry entering along Bangladesh's eastern frontier, additional Iranian and Central Asian ancestry entering along Pakistan's western and northern frontier, and layered on top of all of it, many hundreds of distinct endogamous communities, each carrying their own specific founder population signature, shaped by centuries of marriage within a relatively narrow social and religious boundary. The modern political borders separating these three countries, by contrast, are barely more than 75 years old in the case of India and Pakistan, and just over 50 years old in the case of Bangladesh, a genetic instant compared to the timescale over which the underlying ancestral structure was actually formed, layer by layer, over many thousands of years of gradual change. This also explains why genetic research across the subcontinent has become such a valuable resource for medical researchers, specifically because so many distinct, well-defined founder populations exist across India, Pakistan, and Bangladesh combined, each carrying its own specific genetic signature shaped by centuries of relatively isolated marriage practice. This region offers researchers an extraordinarily detailed natural laboratory for studying how specific inherited genetic conditions spread and concentrate within defined populations over time. Medical researchers have used this data to identify specific genetic variants linked to elevated disease risk within particular communities, informing genetic screening and counseling programs designed specifically around a given community's documented ancestral and marital history, rather than relying on broader regional or national health data that would miss this level of specific genetic detail entirely. Stepping back from all of this, what makes this comparison so genuinely compelling is that it reveals just how disconnected genetic history and modern political history can actually become, even across regions that share an enormous amount of cultural and historical continuity on the surface. India, Pakistan, and Bangladesh are three separate modern nations, each with its own government, its own national identity, and its own distinct modern history, particularly following the upheaval of 1947 and 1971. But underneath all of that recent political history, the genetic structure of the people living across these three countries tells an entirely different story, one shaped by migrations that occurred thousands of years before any of these borders existed, and by marriage patterns within specific communities that have remained remarkably stable across many centuries, regardless of which government happened to be drawing borders through the region at any given point in more recent history. This is ultimately why comparing DNA across India, Pakistan, and Bangladesh produces results that genuinely surprise people, including many researchers who specialize in this exact field. The comparison does not reveal three cleanly separated national populations, each defined by its own distinct genetic identity. It reveals a single, enormously complex ancestral landscape, shaped by ancient migration corridors, centuries of endogamous community structure, and regional geography, all of it layered together over thousands of years, with the modern national borders sitting on top of that deep genetic structure almost as an afterthought, drawn far too recently in historical terms to have meaningfully reshaped the ancestry of the people living on either side of them. It is worth remembering that this kind of research keeps expanding as more communities across the subcontinent participate in detailed genetic studies. Many smaller and less extensively studied communities across rural India, Pakistan, and Bangladesh have only recently begun receiving the same level of detailed genetic attention already given to larger, more accessible population groups. And researchers expect that continued study will keep revealing additional layers of this already remarkably complex ancestral picture. Every additional community sampled adds further detail to a genetic map that, even after decades of research, is still very much a work in progress, reflecting one of the most genetically diverse and historically layered regions found anywhere on the entire planet. It is also worth putting this research in perspective against similar studies conducted in other parts of the world, because doing so highlights just how exceptional South Asia's genetic diversity really is on a global scale. Comparable population genetic surveys conducted across the entire European continent, spanning dozens of countries and hundreds of millions of people, have generally identified far fewer distinct founder populations than researchers have documented within India alone. This is not because European population history is somehow simpler or less eventful, but because European populations have historically practiced endogamy on a smaller scale and with less consistency, across such an enormous number of distinct, clearly defined communities compared to the pattern found across the Indian subcontinent, where caste and community-based marriage practices created literally hundreds of separate, genetically distinguishable founder populations, many of which have remained relatively distinct from each other for well over 1,000 years. This comparison is part of why South Asia has become such a significant focus for population geneticists, specifically interested in founder effects and endogamy-driven genetic structure, since few other regions in the world offer such a dense concentration of clearly documented, genetically distinct communities living in such close geographic proximity to each other. This finding gives researchers a useful, well-documented benchmark for understanding endogamy-driven population structure more broadly, and it is a good example of how a specific regional study can end up reshaping assumptions held across the wider field of human population genetics as a whole. If this kind of deep dive into ancestry and population genetics is exactly the sort of mystery you want more of, this channel is built specifically around uncovering stories like this one, sitting at the intersection of ancient migration, modern political history, and cutting-edge genetic science. Subscribe so you do not miss our next investigation, where we trace a completely different population whose DNA tells a story that contradicts almost everything historians long assumed. And if you found this breakdown useful, let us know in the comments which region or population you would like us to investigate next, since these comments genuinely shape what we research and bring to this channel going forward. And if your own family history traces back to any part of this region, whether in India, Pakistan, or Bangladesh, or to a community that ended up displaced or divided by the events of 1947 or 1971, it is worth remembering that whatever border your family happens to live behind today, the deeper genetic story connecting you to neighboring communities on the other side of that line very likely runs back thousands of years further than the border itself ever has. Political history moves quickly, sometimes redrawing an entire region's borders and displacing millions of people within the span of a single generation. But the deep genetic history layered underneath that political history moves on an entirely different timescale, one shaped gradually over thousands of years, largely indifferent to whatever borders happen to be drawn across it at any given moment.